Hello, I'm Hannah Menke, and I'll be talking to you about our four-dimensional fast tomography study of pore-scale reactive transport in two heterogeneous carbonates and a few of our results. Storing carbon in oil reservoirs has the added benefit of mobilizing more oil for extraction while mitigating climate change. In fact, over 50% of world oil is stored in carbonate reservoirs. Now, as you all know, when CO2 is mixed with water, it becomes carbonic acid, and when acid comes into contact with carbonate, it can dissolve the surrounding pore structure by a measurable amount. This dissolution can, in turn, increase the number and size of permeable pathways for oil to be extracted from the reservoir and significantly affect CO2 storage and capacity and permanence. However, all dissolution is not created equal and is in fact condition dependent. When and how this dissolution happens directly dictates how fluid flow changes in the porous media, and these changes must be fully understood if we hope to upscale from the pore scale to the reservoir scale and make accurate predictions of flow and transport. The way we visualize flow properties is with the Dom-Kohler-Peclet space. The x-axis is flow rate, represented by the Peclet number, and the y-axis is reaction rate, or Dom-Kohler number, which must then be multiplied by the Peclet number in order to isolate reaction from advection and make the axes independent. When flow rate is very high, dissolution is advection dominated, and we see uniform dissolution. When flow rate is low and reaction is high, the dissolution is diffusion dominated, and we see the front of the rock dissolved. This is called compact or face dissolution. And when both flow and reaction rates are high, the dissolution is dominated by preferential flow paths, and we see wormhole formation. The placement and slope of these boundary lines are dependent on the intrinsic rock properties, be that composition, permeability, heterogeneity, etc. We designed this experiment to simulate CO2 saturated brine injection very close to the wellbore, at around 1 kilometer in aquifer depth. We chose two different limestone carbonates, both with a heterogeneous pore structure and two different flow rates. The flow rates were both relatively fast to encourage fully mixed fluids in the pore space, and we used the highest reaction rate possible under these high temperature and pressure conditions by equilibrating our brine with CO2 at 50 degrees centigrade and 100 bar, with no added buffers, which works out to be a pH of 3.1. We then pumped this fluid through each of our 4 rock samples for 2 hours and took between 40 and 90 images during reaction. We chose 2 carbonate limestones for this benchmark study, Estiolatus and Portland basebed. Portland is an oolite that has a heterogeneous pore structure that is poorly connected with a widely varying grain size. Estiolatus is also heterogeneous, but is well connected with different grain shapes and sizes. It is important to note that both samples are chemically homogeneous, being at least 97% calcite with the remaining fraction. So here is our core next to a British penny, which for all you Americans out there is about the same size as an American penny. It is 4 millimeters in diameter and around 1.2 centimeters long. We first wrap it in layers of aluminum foil to stop diffusion of CO2 out of the core. We then put it in a Viton sleeve and snugly fit steel end fittings with PEEK tubing attached and a thermocouple with an additional layer of adhesive aluminum foil. This is then placed inside a carbon fiber Hassler cell and mounted to the rotation stage inside the beamline. Previous lab-based methods made it possible to image homogeneous rocks at relatively slow reaction rates. However, if we want to image more complex rocks at higher spatial and temporal resolutions, we need a more powerful X-ray beam, so we travel to Diamond Light Source. The beamline uses more sophisticated imaging equipment, and the beam is far more intense. Almost 3 orders of magnitude more intense, in fact. This is a picture of the X-ray beam taken using a visible light camera. Why can we see it? Because it is so powerful that it is ionizing the air. Our experimental setup is as follows. CO2 is loaded into the front pressure pump and pressurized. This CO2 is then equilibrated with brine in the reactor at high temperature and pressure. The core is confined at a higher pressure than the reactor in order to control the exterior boundary conditions of the core. A thermocouple inserted into a radial port of the cell is paired with flexible heating tape in order to keep the cell at reactor temperature. Fluid is then pulled through the core for a set amount of time. The carbon fiber sleeve of the cell and flexible tubing allows for images to be taken while dissolution is happening. The post-processing for this dataset is a multi-step process because the dissolution is focused in a few places and happens quickly enough to blur the edges of the rock, making a traditional watershed segmentation extremely difficult. So we register every image in the time series to the first image and then subtract to get the difference image. This can then easily be filtered and segmented and finally subtracted from the first segmented unreacted image to get a time series of segmented reacted images. Here we see the porosity profiles of each of the 4 samples averaged over each 2-dimensional slice of the 3-dimensional image along the axis of flow for each experiment at 30-minute intervals. In all 4 experiments, porosity changed uniformly in the direction of flow. For experiment 1, the porosity also increased uniformly through time. However, in experiments 2, 3, and 4, the majority of the porosity increase occurred during the first 30 minutes with a slower rise at later times. Experiments 1 and 3 had a total increase in porosity of 6.6 and 5.5% respectively, which is smaller than the total porosity increase in the higher rate experiments 2 and 4 of 11.5 and 12.5%. Here, we examine the dynamic changes in porosity and permeability. The change in total image porosity is plotted as a function of time. Experiment 1 had a steadily increasing porosity through the experiment. Experiments 2, 3, and 4 increased in porosity quickly at first and then more slowly. Permeability increases rapidly as a function of porosity. Permeability was calculated directly on the segmented images using the Navier-Stokes solver that computes voxel-centered velocities. The permeability was found from the ratio between the total flow rate across the whole sample to the average pressure gradient multiplied by fluid viscosity from Darcy's law. The greatest change in permeability with porosity was in experiment 4, which had the highest flow rate and the lowest initial permeability. The low flow rate experiments 1 and 3 showed the least change in permeability with porosity, while experiment 2, which had a very high initial permeability, had large changes in porosity with a smaller permeability increase. We fit the data to an empirical power law relationship between porosity and permeability. The best fit to the Estiolatus experiments 1 and 2 were exponents of 6.7 and 7.5, respectively. For the Portland experiments 3 and 4, we found exponents of 10.9 and 11, respectively. The exponents, around 6 to 11, are larger than the value of approximately 5 observed for dissolution of a homogeneous limestone keton, observed in Menke et al. 2015, and much higher than a power law of approximately 3 used in the Kozeny-Kármán relation to predict the permeability of granular packings. This suggests that we see a very rapid rise in permeability. The specific surface area, S, was computed on the images by adding the number of voxel faces separating grain from void. S is the surface area, A, divided by the bulk total volume. S showed an approximately linear increase with time for experiment 1 and a sharp initial increase for experiments 2, 3, and 4 with less change at later times. Here, we visualize the ratios of the magnitude of velocity U at the voxel centers divided by the average pore velocity for Portland and Estiolatus carbonate. U average is calculated from the Darcy velocity divided by the scan porosity taken from the segmented image. The Darcy velocity is constant throughout each experiment. It is imposed by the flow rate of the pumps. Here we see that the initial flow is directed through small preferential flow paths in experiments 2, 3, and 4, while there are many initial flow paths in experiment 1. This is confirmed by the initial permeability for all experiments except experiment 1. When we look at the PDFs of velocity, we see 2 distinct regions for Portland of stagnant velocities on the left and advective flow on the right. The same is true for Estiolatus in experiment 2 with a smaller stagnant region, while in experiment 1 the sample is dominated by advective flow with very few regions of stagnant flow. 60 minutes into the experiment, we observed that the initial preferential flow paths have widened in all experiments. Furthermore, the permeability has increased substantially for experiments 2 to 4, but only marginally for experiment 1, which had an initial permeability that is much larger than the other samples. Furthermore, when we look at the PDFs of velocity, we see that the velocity distributions are almost identical to the initial for experiment 1. However, in experiments 2 to 4, we see that the advection has become dominant and that many of the stagnant regions have disappeared. At the end of the experiment, we observed that the initial preferential flow paths have widened a little bit more in all of the experiments. Furthermore, the permeability has increased marginally for all experiments. Additionally, when we look at the PDFs of velocity, we see that the velocity distributions are still almost identical for experiment 1, with a peak velocity slightly lower to account for the increased porosity. In experiments 2 to 4, we see only a marginal change, and that advection has remained dominant. Interestingly, we also see the emergence of a third peak, which could indicate competition between preferential flow paths. The most significant feature is that the reaction rates are much lower than the values measured on a flat surface with no transport limitations. Initially, the effective rates are at least 10 times lower, declining to 100 times lower at later times. This demonstrates that reaction is severely limited by the rate of transport of reactants to the solid surface. When channels form, these dominate the flow and limit reaction to the sides of the channels themselves. In comparison, the transport of reactant and products in the pore space away from these channels is very slow. This discrepancy between intrinsic and effective rates is larger than the 14-fold variation observed in the more homogeneous Ketten sample in Menke et al. 2015. This is a schematic of uniform dissolution, where we see the rock in white, pore space in black, and the presence of reactive fluid in blue. In experiment 1, we observed uniform dissolution, which is what we would expect in a low reaction rate, high flow rate conditions in a well-connected rock. However, in experiments 2 to 4, we observed something quite different. This is a schematic of traditional definition of wormhole formation, where the reactive fluid front just precedes the pathway that is opened by the reactive fluid. This is what we would expect according to traditional reservoir engineering, when the Dom-Kohler number is high, as it would be with a very acidic solution such as hydrochloric acid or EDTA. So while the fluid is directed down the preferential flow paths, the dissolution is fast, and the preferential flow paths are dissolved in steps. However, during our experiments, we found a new dissolution regime that we call channel formation. During channel formation, flow is faster than in wormholing regime. Reactive brine floods the rock very quickly, but the reaction of the CO2 saturated brine is slower than a strong acid, and the heterogeneity of the rock allows preferential flow paths to form, which then provide the majority of the reactant to a more limited portion of the rock surface. This means that the effective reaction rates in the channeling regime are up to 2 orders of magnitude smaller than that measured on a flat substrate with no transport limitations, implying that transport of reactant and products in the regions of the pore space away from the channels is significantly impeded. Furthermore, the initial connectivity and permeability of the rock dictates how many channels exist and which channels will be preferentially dissolved. If there are many channels that are well connected to each other, then the rock will be dissolved uniformly, while if there are only a few poorly connected channels, then we expect to see channels form. To conclude, we have found that there is a clear impact of initial structure on the changes in porosity and permeability due to fluid-solid reaction in carbonate rocks. We see the emergence of a new type of dissolution pattern in heterogeneous systems, channeling, where preferential flow paths widen and dominate the flow and the dissolution process. The effective reaction rates in the channeling regime are up to 2 orders of magnitude smaller than that measured on flat substrates with no transport limitations, implying that transport of reactants and products in regions of the pore space away from channels is significantly impeded. This paper is now published and freely available in the journal Chemical Geology. Furthermore, the datasets are available for download on the website of the British Geological Survey, and any code used for image processing is available on GitHub. I'd like to thank our sponsors and acknowledge our references. Also, special thanks to my colleagues at Imperial College and the Diamond Lloyd Search team who were essential in setting up these experiments. If you have any questions, feel free to comment on this video or to contact me on my website, hannahmenke.com, or on LinkedIn.